my name is Macy Steber. I'm a junior at Holt High School, and my project looked at the effects of vitamin D3 on glucose absorption in Drosophila melanogaster DFDLP1 through 5. So type 1 diabetes is an autoimmune disease that causes the body to attack the insulin-producing beta cells that are located within the pancreas. Over 1.5 million people in the US alone live with type 1 diabetes. Now, life with type 1 diabetes entails a life of closely monitoring blood sugar levels, accurately counting carbohydrates, and injecting insulin. Blood sugar levels are affected by anything from stress to diet to exercise and to so many other things. Now, not maintaining good blood sugar levels can lead to other health problems, so it is important for diabetics to maintain good blood sugar levels. Now, I myself am a type 1 diabetic, and I personally know the struggles of trying to maintain good blood sugar levels. There are methods and devices out there to help type 1 diabetics in maintaining good blood sugar levels. However, these are often relatively expensive and are not readily available. I wanted to find a way that is cheaper and more accessible to help those who are struggling to maintain good blood sugar levels. And a while back, I was put on vitamin D supplementation, and I noticed that my blood sugar levels were getting easier to control and were in a better range. So I wasn't sure if this was due to the extra vitamin D or if it was because I was becoming healthier. So from this, I wanted to see if vitamin D could potentially be a way to assist in maintaining good blood sugar levels for type 1 diabetics. So vitamin D is acquired through either diet or through sunlight. Tissues have been found to express 1-alpha hydroxylase, which activates vitamin D3. Now, this suggests that vitamin D can act in a paracrine or autocrine manner. Now, vitamin D deficiency has been linked to the onset of several autoimmune diseases, such as type 2 diabetes. Now, not much research has been done on vitamin D and type 1 diabetes. However, there was a systematic review done that showed that vitamin D has a short-term positive effect on individuals who are newly diagnosed with type 1 diabetes. There's also a study done in Italy on an 8-year-old child where they were given doses of vitamin D3 and omega-3 fatty acids at the time of their diagnosis. A year later, they were able to maintain good blood sugar levels with only 1.5 to 2 units of insulin per day, which is substantially lower compared to what a typical diabetic uses. So, fruit flies or Drosophila melanogaster share about 60% of their genes with mammals. Now, through the deletion of an insulin-like protein, Drosophila become a suitable model for type 1 diabetes and show the diabetic phenotype. Now, the 60% of genes that are shared result in similar metabolic and signal transduction pathways, which make them a suitable model for type 1 diabetes. So to make all this information about vitamin D and type 1 diabetes more applicable to myself and to others who are not newly diagnosed, I asked the question of how will varying levels of vitamin D3 affect glucose absorption in Drosophila DFDLP1 through 5. So it is hoped to be concluded from this project that higher levels of vitamin D3 will increase the efficiency of glucose absorption in Drosophila DFDLP1 through 5. So to look into this, 4 cultures of the Drosophila were set up. The first one was a control group and was given no vitamin D supplementation. The first group was given 63 microliters, the second 126, and the fourth was given 189 microliters of vitamin D. Now, this vitamin D was mixed into the media that the fruit flies were kept on. So they were kept on this for 10 days, and on the 10th day, they were starved, and then they were injected with about 1/10 of a microliter of a PBS insulin solution. After this, they were allowed to recover for either 15 or 30 minutes, and then they underwent a hemolymph extraction process where their hemolymph was collected. Now, from this hemolymph, the glucose concentrations were determined. So as you can see here, these are the glucose concentrations. And after a one-way ANOVA was run, it was found that there was no statistical significance in my data. However, as can be seen here, the group that was supplemented with 63 microliters resulted in lower overall glucose concentrations, and the group that was supplemented with 126 resulted in a decrease of glucose levels over time, which is not seen in any other group. So this leads me to believe that there may be a correlation between vitamin D3 and type 1 diabetes and glucose concentrations. So for my experiment, this was only one trial, so it is possible that the data is not accurately representing what would actually happen. It is also possible that the PBS insulin solution does not have a similar effect on flies as the insulin-like proteins do that the flies have. It is also possible that the vitamin D could have affected other parts of the fly and ultimately made the fly unhealthy. So for the future, I would like to conduct more trials to see if what I saw can be replicated. I'd also like to conduct trials on wild-type flies to see if vitamin D plays a role in the glucose absorption in non-diabetics. I'd also like to look into the exact effects of vitamin D3 on the signal transduction pathways with insulin. And finally, I would like to see if what I'm seeing in the fruit flies, if that can be replicated in humans. So overall, vitamin D3 has the potential to have positive effects on glucose absorption in Drosophila melanogaster DF-DILP 1 through 5. So I would like to thank Paula Kiefel from Washington University in St. Louis, Dr. Leslie Pick from the University of Maryland, and Ms. Jennifer Hess. Thank you. Are there any questions?
Yes, you referenced vitamin D and using vitamin D3. Are these typical over-the-counter products? I was wondering if concentrations had a high enough percentage compared to the average dosages that people might receive.
Yes, so the vitamin D that I used was a gel from a pill, so I just took it that was in it. So it was just your typical over-the-counter vitamin D that was used in this project.
Okay. And why did you starve the fruit flies before the insulin injection?
So they were starved, and then I forgot to mention it, but then they were fed a 10% glucose solution. So it acts kind of like how regular people would eat, how you would eat, and then you would inject yourself with insulin.
Okay. Kind of emulating like a real human interaction.
Yeah.
All right. Wonderful. Thank you very much, Macy.